Hello everyone. So today the topic is the operon. So the operon part will be covered in your class 12th, as well as if you are preparing for any competitive exam, like if you are preparing for CSIR or any other exam, it will be beneficial for you. In NEET also the operon will be beneficial, and if you are a student of BSc or MSc, you can use this notes, because I also use this particular notes when I was writing my bachelor's and my master's exam. So, the gene regulation in prokaryotes and eukaryotes take place by different method. In eukaryotes, the gene regulation takes place by chromatin remodeling, which is a separate topic, okay, which we will discuss later. But today the topic will be operon. Operon are basically the gene regulation in prokaryotes. Like operon is basically a method of gene regulation in prokaryotes. Okay, so it is basically multicystonic. Now, operons there are two type of genes. The first genes are which are always on, and the genes which are always on are known as housekeeping genes, and they are consecutively expressed in the operon. It means if they are on, they will be on. You cannot switch it, like you cannot off and on it. It will be on regularly. Okay. And then we have regulatory genes. So means you can switch this. Like if they are on, you can also convert them into off situation. And if they are off, you can convert it into on situation. Okay. Now in operon, we have two type of operon. First is inducible and the next one is repressor. Inducible means you can induce them, like you can on them. Inducible means you can on them. So if they are on, you can on them. Repressor means you are repressing them. Means that you are making them in a rest situation. Like you are off, you are doing them off. So basically, if they're in on situation, it will get converted into off situation. Okay. Now in operon, we have three things. Mainly we have three things: promoter, operator, and structural gene. Here P stands for promoter, O stands for operator, and then we have structural genes. So this is the basic gene regulation, like operon, which I also have written in my exams. And now we come to the next part. Okay. So we have promoter, operator, and structural gene. Now structural gene are basically, you can here I am writing the X, Y, Z. So I just have gave name to these structural gene X, Y, Z. Operon basic meaning is like when one gene is transcribing under single promoter, it is basically an operon. It means that you only have a single promoter, and more in a single promoter more than one structural gene are getting transcribed. Okay. So it doesn't mean that operons are not present in eukaryotes. Like operons are present in both prokaryotes as well as eukaryotes, but they are mainly present in prokaryotes. Okay. Next part we have. Okay. So if we are talking about activator protein, activator always stands for positive regulation. Activator always stands for positive regulation, and repressor always stands for negative regulation. Negative regulation. So activator means positive regulation, repressor means negative regulation. Okay. So here the plus stands for the on situation. So if they are off, you will convert them into on. Minus stands for minus stands for repressor, and positive stands for activator. Okay. The positive here is the activator, the negative here is the repressor. Okay. And then we have I means inducible. Inducible means you are making the operon in the on situation. Okay. So if they are off, they will be on, because they are inducible now. I means inducible, you are inducing them, you are making them active, and negative means repressor or positive means activator. Same situation here also. R stands like you are repressing them, means you are converting them into the off situation, and positive here means activator, negative here means repressor. Now a very important part is that active, if the protein is activator, the activator will always bind to the promoter, will always bind to the promoter, and if the protein is repressor, it will always bind to the operator. You need to remember this. If the protein is activator, it will get bind to the promoter. If the protein is repressor, it will get bind to operator. I will provide you notes if you want it, but I am telling you this is the best notes of operon you will ever have. Either return part, either learning part, either understanding part. I think this is the best notes of operon you will ever have, because I have solved every competitive exam questions from this particular notes of operon. CSIR net, you can solve any questions from this notes, and you can also write these notes in your BSc as well as in MSc. Today we will only discuss about lac operon part, but in the next video we will also cover tryptophan, histidine, and other such operon. So today we will only discuss about lac operon. Okay, let's move further. Now we have two type of operon. The first type is catabolic and the other type is anabolic. What do you mean by catabolic? Basic. Catabolic means if you have large things, you are breaking them into the small things. Anabolic means you are joining small things and making them a larger thing. Okay, I am just sharing with you all a trick. CI, you have to remember that CI. If the CI is here, I means you are inducing them. It means that you're converting the operon into the on situation. Okay, so you have to remember it by CI. Catabolic, inducible. Okay, so here it is written also, catabolic follows inducible system. Example of the catabolic operon is loni, toni and oni. Lac operon, trehalose and arabinose. Loni, toni, oni. Lac operon, trehalose and arabinose. Clear? This is the slide of catabolic. If you want to write the notes, you can screenshot, you can take the screenshot also. Then we have anabolic, opposite. Agar CI ho gaya, then we have AR. Anabolic means repressible. So you are joining the small things and you are making it a large. Okay. The example of anabolic is tryptophan and histidine. Okay, I just forget one thing to tell you, that the main operon here from the lac, trehalose and arabinose is only the lac. And lac operon follows minus R situation. Lac operon follows minus I situation. Minus means repressor, I means inducible. So you are converting the operon into the positive situation. You are basically converting the operon into the active operon. Okay, you are converting situation into the on situation. Then we have tryptophan and histidine, which are the example of anabolic. So they both are minus R and minus R. Means minus means repressor, R also means repressor. Clear? I hope that much is clear. Okay, today we will only discuss about lac operon. Otherwise this video will be very very large. So tryptophan and histidine operon I will try to cover later. Tryptophan is more important, histidine is not that much important, but in MSc part you have to learn about histidine. I think in BSc part histidine is not included, only the lac operon and tryptophan is important. One thing I want to tell you, if you are preparing for CSIR NET, you don't have to remember histidine, because the much I know, histidine is not included in CSIR NET syllabus, only the lac operon and tryptophan is included. Okay. Now let's come to the lac operon part. Lac operon is present in mostly prokaryotes. Okay, E. coli example is this. So lac operon is given by Jacob and Monod, and I tell you that it is minus I. Minus means repressor, I means inducible. Okay, so in our body also lactose sugar is present. You all know about the lactose, right? Lactose is basically a milk sugar. It is a B galactosidase that E. coli use for energy. So basically E. coli consume the lactose for energy and also as a carbon source. Like E. coli use lactose as a carbon source as well as for the energy source, because Basically it's a food for E. coli. E. coli consume lactose in the form of a food. Okay. Now in our body also lactose get converted into glucose plus galactose. मतलब lactase की presence में it will get converted into glucose and galactose. Okay. In lac operon we also have promoter, operator and structural gene. Here I represent the repressor protein. So I get transcribed, get translated, and then it will form repressor protein. As I told you, repressor protein will always bind to operator. It will not bind to activator. So now repressor protein is getting binded to the operator. Now if the repressor protein gets bind to operator, so obviously it is like it's a very obvious thing that if the repressor protein is getting bind to the operon, then how it will be active? The gene will get off. This is very obvious. Now, if a repressor protein is binding to operon, it will get off. Clear? I hope this much is clear. I think you cannot make it that much because I think operon is typical part. It's not a easy thing. I hope you are getting it. Okay. Let's move further. Okay. I told you na, I means repressor protein. It get transcribed, it get translated, it will form repressor protein. Now repressor protein is active. If the repressor protein gets bind to operator, then genes will not get transcribed. Okay, then that B galactosidase, permeases and this will never be produced. Okay, but it's an inducible operon. What do you mean by inducible? Induce हो रहा है, which means you are making it active. It means on is converted into off is converted into on. So first situation was this, because initially it was off. After it will convert it into plus I, like on हो जाएगा, it will get on. How it will get on? So we will learn this. So the thing is, on the E. coli membrane, some permeases are present, and these permeases allow lactose to enter into the body of E. coli. Now, if the lactose is entering from these permeases, it will get converted, either it will get converted into glucose and galactose, or it will get converted into allolactose. Okay, so the permeases are coming through this the lactose is coming through permeases and it will get converted into allolactose plus, or we can call it glucose or galactose. Okay, the difference between lactose and allolactose is, in lactose glucose and galactose are binded with beta 1,4 bond, and in allolactose glucose and galactose are binded with beta 1,6 glycosidic bond. Very easy, you just need to learn it. Nothing to understand here. You need to learn it that in lactose B14, like beta 1,4 bond is present, allolactose beta 1,6 bond is present. Okay, now come further. Now we know that initially in E. coli lactose was absent. Okay, but through the permeases lactose enters into the E. coli. Now the lactose will get bind to the repressor protein. Now lactose will bind to the repressor protein. Now the lactose, because the lactose binded to the repressor protein, now this is known as allolactose. Now it is known as lactose, like repressor protein gets binded to the lactose, or we can call it allolactose. Like, it's not compulsory. Dekho, like only the lactose bind the repressor protein, है ना? So when the lactose bind to the repressor protein, initially the repressor protein was active. Now it will convert it into inactive. And if it is inactive, how it is possible to bind to operator? So the repressor protein will not bind to the operator. And because the repressor protein is not binded to the operator, it will be on. So that structural gene, which is XZYA, or what we call XYZ, is not like wo. So the structural gene which are present in the lac operon will get transcribed and it will get converted into beta galactosidase permeases and trans-acetylase. Are you getting me? If the repressor protein gets bind to lactose, or what we call allolactose, it will be not able to bind to operator, and the genes, the structural gene which are present in the lac operon will get transcribed, and it will be converted into the like B-galactosidases, permeases, and trans-acetylase. So these are the three structural gene of lac operon. You can screenshot this diagram. This, if you make this diagram in your BSc exam sheet in your papers, you will definitely get marks, because I also get marks from this diagram only. Okay, now let's come to the next part. LAC-Z codes for beta galactosidase. Beta galactosidase के पास में, like the beta galactosidase have this power of cleavage. Cleavage means it will get broken. Like beta galactosidase is capable of breaking lactose molecule, and when the lactose molecule breaks, it will form glucose and galactose. Okay. So either it can form glucose or galactose after the cleavage, or the lactose without cleavage will get converted into the allolactose, and this particular function is played by beta galactosidase, which is coded by LAC-Z. LAC-Z is one of the structural gene of operon. Okay. Wild type means that initially the E. coli do not have beta galactosidase, because there was no lactose. Initially the lactose was absent in the E. coli. We have learned it that initially lactose was absent, that why repressive protein was getting bind to the operator. But after some time, when you provided the medium of the E. coli, lactose, then the concentration of beta galactosidase increases. Wild type means that initially. Okay, I hope this is clear. You want to take a screenshot, you can take. Okay. LAC-Y codes for beta galactosidase permeases, which function is to transport lactose inside the E. coli. Like beta galactosidase permeases make the lactose permeable, like it allows the entry of lactose into the E. coli. LAC-A codes for beta galactosidase acetylase, like LAC-A codes for beta galactosidase acetylase, or what we called as beta galactosidase trans-acetylase. Okay, beta galactosidase acetylase, or what we call beta galactosidase permeases, acetylase or trans-acetylase. These two this structural gene basically not play an essential role in lactose metabolism. The main role it plays was in detoxification. Okay, so it do not play any role in lac operon, या फिर little bit role in lac operon, but the thing is कि it play a role in detoxification. Okay. So, lactose is actually an inducer of lac operon, because it is inducing the operon, because when you growing the E. coli in the wild medium, it was not able to get expressed, like structural gene was not able to get expressed. But when you add allolactose in that particular medium, the thing is that lac operon started transcribing its structural gene. So lactose is the inducer of lac operon, but allolactose is also an inducer of lac operon. Other than allolactose or lactose, isopropyl thiobeta galactosidase, which is a synthetic non-metabolite of lactose, it is also an example of, it is a synthetic inducer. Like lactose and allolactose are not synthetic, but the thing is isopropyl thiobeta galactosidase, it is a synthetic metabolite of lac operon. It is also an example of gratuitous inducer. Gratuitous means it is continuously expressing in the operon. You want to take a screenshot, you can take. This is how the lac operon regulation takes place negatively. So, okay, now in BSc, when you are BSc or MSc, there is a question comes that is negative regulation of lac operon. So, you can write jitna, the part I have taught you, until now, you can write this particular in the negative regulation of lac operon. This question comes, okay, negative regulation of lac operon. The positive regulation we will discuss in the next video. The positive regulation is also known as the catabolic repression, okay, this part. So I will make you understand this part in the next video. Till then, this is like, this part is like, you can write this part in, if the question is like you have to explain the lac operon, you can write this. If the question is negative regulation of lac operon, you can write this. If the question is you have to write something about operon, you can write this. Okay, so if you are BSc MSc student, it will be beneficial for you, and if you are a student of class 12th boards, you can definitely write it. If you write some extra points other than your NCRT, your marks will not get deducted. It will give you good marks. So yes, this is a negative regulation of lac operon, or what we call the short note on lac operon, or operon also. Okay, thank you. Next video we will discuss about the positive regulation of lac operon.